So it's my great pleasure to be here, and I thank the organizer for inviting me here to the very important meeting. So in the last decade, we have seen many good progress implementing the pharmacogenomics guided drug development and other administrations. But we still have some challenges to further promoting the pharmacogenomics in the clinical practices. So today I would like to go to introduce some of the example focusing on the ethnic differences between Japanese and other populations, and followed by showing a couple of challenges so we need to consider. And first example is the carbamazepine-induced Stevens-Johnson syndrome 10 associated with HLA-B1502. So that is already mentioned yesterday by a couple of speakers. And then now, as you know that HLA-B1502 screening could provide a benefit in the country in which HLA-B1502 is relatively prevalent, such as Han Chinese and the Thai populations. And Dr. Chen nicely showed that and published in the New England Journal of Medicine, and HLA-B1502 screening will reduce the risk of Stevens-Johnson syndrome caused by the carbamazepine. However, the carbamazepine-induced Stevens-Johnson and 10 patient carrying HLA-B1502 have not actually been found in Japanese, so reported by the couple of Japanese researchers. Instead, so recently in Japanese, association with a different allele, HLA-A 3101, has been reported by the Ozeki, Tokyo Riken groups. And more interestingly, and similar results were found in the European populations. So the European population also have some association with HLA-A 3101 and the same allele to the Japanese populations. And now this is a current level of the carbamazepine in Japan. And you may not read the Japanese label, but you can clearly see here the HLA-A3101 and here the HLA-B1502. We include both scientific fact in the Japanese label, saying that the HLA-A3101 is associated with carbamazepine-induced serious cutaneous adverse event, including SLS10. And we also describe the HLA-B1502 is associated in Han Chinese, but not in Japanese populations. And furthermore, clinical meaningfulness of the HLA-A3101 on patient selection is still unknown. We need the more data. Actually, prospective study in Japanese population is undergoing to reveal this important issue. I'm looking forward to the data in the near futures. And other example, here is the GEF chamber and the EGFR mutation, and the higher EGFR mutation rate as in Asian population, including Japanese, reported, and 30 to 40% to whole Asian, and 5 to 10% for non-Asian populations. And prospective study in Japanese populations confirmed higher efficacy of the GEF chain in non-small cell lung cancer patient with EGFR mutation. And some other clinical trial also confirmed the same result and the evidences. Therefore, now the indication for this drug is limited to the EGFR positive patient only. And pre-screening for the EGFR genomic testing is required before starting GIF genomic therapy in Japan. And those testing fees is also covered by the National Health Insurance in Japan. And the final example is the irinotecan and UGT1A1 allele. As you may know, the STAR28 is a common allele among the African American, Caucasian, and Japanese populations. But for the STAR6, it's only found in the Japanese population, approximately 17%. That is a substantial number in the populations. And the prospective study confirmed the higher risk of the neutropenia caused by the irinotecan in patients with heterozygote and the homozygote population of STAR6 or STAR28 allele. So therefore, the label of this drug in Japan describe the higher risk may cause in the population with these alleles as important information. And then also this screening testing the in beta assays are available and then will be covered by the National Health Insurance. And I also show you some of the recent research results we conducted. So we compare the PGX biomarker information and how that information is described in the drug level between US and Japan. So we collected biomarker information published in the US FDA website and then compare the contents and the sections what is the grading level of that information for the precaution. And actually here you can see the 5 biomarker statement is included in the warning section in US level, but only 1 of the biomarker is mentioned in the warning section of the Japanese level. And similarly, and 23 biomarker as mentioned in the indication doses in US, but of those 14 biomarker information is included in the Japanese level. Actually, 19 biomarker information is not mentioned in the Japanese level. So we go into the why such differences happened. And then here's some of the actual example. Here, first one is a warfarin, the CYP2C9 as a biomarker. That's included indication usage section of US level. But in Japan, that information is included in the clinical pharmacology section as a reference information only, without any practical guidance for the dosage adjustment. And as I described earlier, HLB 1502 is also included in box warning in US, but in Japan, that information is recognized as other precaution sections. And the DPD for the campistobin, that's also included in control indication section in US, but in other precaution in Japan. So this is a possible reason for the differences. Maybe some of the clinical evidence in Japanese is not sufficient to make a clear statement how to use this biomarker information in clinical practices. And also, as I described, the genetic difference is existed for some of the biomarker. And in addition to the genetic differences, availability of the diagnostic agent, companion diagnostics, is also affect the grading level in the drug level. Here is a more quantitative analysis for that. And the grading, this one is a grading difference shown between the US and Japanese level. This one is for the without grading differences. And for the biomarker, having the grading difference is usually shown for the safety biomarker, and which is mainly for the non-cancer drugs. And those biomarker is usually does not have enough evidence in Japanese population. So that is one of the reasons for the differences. In contrast, so the biomarker with non-grading differences is usually found for the efficacy biomarker, which is mainly for the oncology drug. So, and then those biomarkers they usually have some substantial evidence in the Japanese population. So because for the efficacy biomarker, so that's kind of the biomarker is used in clinical trial before marketing authorization. And that's data is usually included and available for our review before approval. But instead, for the safety biomarker, it's not usually available before marketing, and also it is difficult, not easy, to accumulate the substantial evidence even in the post-marketing stages. And here I describe some of the challenges in PHX-guided drug development and the factor. As you know, so now the drug, almost most of the drug is developed globally, and the multi-region clinical trial is common, and then it's conducted in almost everywhere in the world. And here is a biomarker positive population is quite common in region C, but rare in the region A, and maybe the region B is the intermediate region for that biomarker. But if we conduct a multi-regional clinical trial, those patients for the biomarker positive could be included, enrolled in that MRCTs. So the challenge is how we can evaluate the data in biomarker positive population coming from various regions. So if the drug response is only affected by the genetic factors, answer is yes, but in the reality, so the drug response could be affected by other factors, especially for the extrinsic factor as well described in the ICH-E5. So, but we already have to evaluate this kind of the data for the marketing authorization application. So we quite substantial discussion with industry colleagues how to evaluate this kind of the data. And in the era of the globalization of the drug development, so I encourage the sponsor to use the pharmacogenomics in global clinical trial. And also, I would like to encourage to including the major ethnicity from an early stage of drug development. And we usually also encourage to discuss with the PMDA about the development strategy with PHX before conducting the clinical trial. So important point is that we need to have a grasped impact of the pharmacogenomics in drug responses before conducting the confirmatory trial. How does PGX biomarker is really affect to the drug responses? We need to consider that in including ethnic differences. And here's some of the challenges, and it's the PGX information in the drug level. So further evidence is needed to describe the practical guidance for a safety biomarker, such as dose adjustment and patient selection. So which section or font level warning could be appropriate to describe? So maybe included in the control indication section, or dosage section, or in the clinical pharmacology section as a reference information. And that biomarker it will be required before starting the drug therapy, or just a recommendation, or just information. So how we can clearly standardize such kind of the concept in our regulatory decision making. And also, so in the globalization, so how to describe the PGX information data relating to ethnicities. So personally, in my opinion, so we need to include more information data in a stratified population in terms of race, nationality, or ethnicities. So, how we can describe, and that's information in the drug level, which is very limited spaces, and also how to guide how to use such information by the physician in the clinical practices. So we need to consider that. I don't have a perfect answer for this, but actually, and the very important point is that we need a harmonized approach internationally for that concept. Otherwise, the PGX biomarker information could not be implementable in the clinical practice in a hospital. And finally, I just would like to describe briefly and recent PMDA activity. So here is a summary of the current PMDA scientific consultation. In addition to the standard consultation process, we established special consultation of PGX biomarker qualification, and that's cover from the non-clinical to post-marketing stages. And also, we started the prior assessment consultation that is focusing on the pre-evaluation of the data before formal NDA submitted. That's really useful and shorten the review time for the drug approval in the NDA review stages. And recently, we more strengthen the activity on the pharmaceutical affairs consultation. That means we discuss the drug development strategy itself, how we can make a contribution to the innovative drug or the sheets discovered by the academia or venture company in Japan. So how we can promote such kind of the concept in a whole reality as a whole regulatory approval in Japan. So those kind of the various kind of the scientific consultation program, I believe also they're very useful to promote the personalized medicine in Japan. And recently, we also tried to strengthen our regulatory science research in PMDA. So we published our perspective in Clinical Pharmacology and Therapeutics 2011, and we called the regulatory science bridge, which consists of the three main functions. First one is the tools for data production, such as how to use the biomarker in clinical trial, or how to use the adaptive design for the data evaluation. And the data assessment, second component is the data assessment. What is the meaning of the safety itself? How we can objectively evaluate the safety, and in terms of the risk-benefit balances. And the final point is the balancing of the various factors taken into consideration. So in addition to the benefit-risk balance evaluation, so for the regulatory decision-making, we also take into consideration various kind of factors such as a medical need, social need, and some other risk factors for the maybe the orphan diseases, pediatric diseases, and then how to reduce the off-label use and how to promote the optimal use of the drug based on the this information described in the label. So the many kind of factors should be into consideration for our regulatory decision making. So we believe the stronger, more complete regulatory science bridge will help us for the future drug development, and we can provide the better drug based on the latest science coming into the patient and the societies. And then finally, I would like to propose a smart global concept in the recent globalization of the drug development. We need to collaborate together. And then first one is S is coming for the sharing data, experience, and knowledge globally. And M means the managing the project issues globally, but not by the one company or the one regulatory agency. We need to manage the issues globally for the discussion. And of course, we need to advance the regulatory science, but it's not the US FDA or the EMS matters, and as well as the PMDA matters. We more globalized regulatory agency, as well as the industry clinic, we need to work together to advance the regulatory science, so which could be implementable for, and then can be used for our regulatory decision making. And for the globalization, we need to respect for other idea views globally from various different kind of the perspective. So this workshop is one of those, and we need to learn from others to find out the best way to move forward. And also finally, so we need to be transparent, to establish a transparent process globally. and to promote the understanding the concept of the idea. And then, so we also should be transparent for the public. So we need to explain why the regulatory agency decided this way or this one. So and then we have to communicate with the public. And then those kind of the concept. So I believed if we work together, so that's very helpful for patient, and then we can provide the better drug based on the pharmacogenomics guided drug development to the patient in the world. Thank you very much for your attention.
Thank you very much, Yoshi, and thank you very much for giving us these final guiding principles that I think should be a guide for this whole conference. Could I just, I think we're going to be a little bit short of time later on, so maybe if anybody has questions now, we could take one question. Please.
Thank you for a nice overview, Yoshi. We've had a couple of approvals at the FDA, one in the past couple of years where the drug was not shown to have any benefit in the United States, but had a significant survival advantage in the rest of the world. We've seen, shifting in where clinical trials are being conducted over the past decade or so, to more trials being conducted in South America and Southeast Asia. We also are beginning to have concerns now that with some of these pharmacogenomic findings, for instance, 2C19, to the extent that it could be involved in the activation of a prodrug, or IL-28B to the extent that it affects the hepatitis C background, could shift where trials are being conducted. Do you have any thoughts on sort of the interpretation of global trial results relative to, enrollment in Japan or other Southeast Asian countries?
Yeah, thanks for your question. So that's also happening in Japan. So we published our guidance document in 2007, describing the general principle on the global clinical trial. That guidance document described how to calculate the Japanese sample size to make a consistency among the region in terms of the efficacy mainly. But in the reality, so it's very difficult to include a substantial number of the Japanese patients, and especially for the oncology or some other orphan diseases. And then, so result, we reviewed that as you said, so sometimes the benefit-risk ratio in Japanese population is worse than the overall population, sometimes good. And so we usually discuss the matters, what kind of factor could affect such drug responses. And then but in my opinion, so the more important point before starting that trial, how we can assess the ethnic factor causing the drug response in terms of efficacy. And then, so result is something that they're happening by chance in terms of the statistical powers. And then, so that's why the now happening in the PMDA is we will discuss with the industry colleague what is a possible factor to affect the drug efficacy. But we usually accept the data in the overall population for our regulatory decision making if substantial consideration made before starting the multi-regional clinical trials. And then, so in the future, so we encourage the sponsor to include the Japanese population not only for the confirmatory stages, but from the exploratory stages, more early stage of the clinical trial, to confirm the robustness of the data and confirm the such tendency in Japanese population could be same in the different trials. So if we look at such kind of the situation, probably we are happy to make a decision more appropriately.
And do you have an expectation that sponsors or drug developers would evaluate specific genetic factors that might be at higher prevalence in Asian populations?
And
Should they stratify their results by that, or at least pre-specify that and ensure they have complete data?
yeah, we encourage to use the pharmacogenomics in the clinical trial, and also we encourage the sample collection in clinical trial for the future genetic analysis. But still, it's not so popular to use this kind of the technique, especially for the non-cancer drugs. So we need to work more to promote this concept implemented in other disease area as well.